dear students. Welcome back to the class of sterilization and disinfection. So we have completed the definitions, classification, dry heat methods and moist heat methods. So what we have left with is radiations and filtration. Those two are the physical methods of sterilization that we have to cover in this class. So radiation and filtration. So coming to radiations, so since we have dealt with all the heat methods, so radiation, what the thing which comes to your mind when you hear about radiation is the UV rays, X-rays, gamma rays and infrared rays, right? So we can divide radiations into two types, that is ionizing radiation and non-ionizing radiation. Okay, so hearing this, which will be the better method? It is definitely ionizing radiations, which are more powerful radiations. So X-rays, gamma rays and the high-speed electrons come under ionizing radiation, and UV light and infrared rays come under the non-ionizing radiation. So what is more important for us, for exam point of view, is the ionizing radiation, and you call this method as cold sterilization. Now this is a previously asked question, 2-mark questions. So what you have to mention when the question is cold sterilization or ionizing radiation, you have to say the radiations which we use here are X-rays and gamma rays. And what do? They are very highly lethal to the bacterial DNA. They can destroy the bacterial DNA. We generally know radiations can cause damage to nucleic acids. And they have very high penetration power. So they're very good sterilizing method. And why it is called as cold sterilization? Because they don't cause increase in temperature. There is no appreciable increase in temperature. That is why it is called as cold method of sterilization. So what kind of articles you can sterilize using this method? Those articles which cannot withstand high temperatures, which may be destroyed by high temperatures, like the plastics, all kind of disposable materials like the swabs, catheters, suture materials, infusion sets, IV cannulas, all those which are coming as a pre-packaged thing, disposable thing. So we put it in this cover and the entire set is being sterilized using cold sterilization. So such kind of articles which cannot be put under a heat method of sterilization, you go for a cold sterilization method where we use ionizing radiations. And what are the ionizing radiations? X-rays and gamma rays. So we use a control, sterilization control here also, like autoclave, Bacillus thermophilus. Please remember the controls: hot air oven, Bacillus subtilis. Here the sterilization control is Bacillus pumilus. And please remember, there is no increase in temperature. That's very important. And what are the radiations we use? X-rays and gamma rays. So radiations, the method of sterilization control we use is a biological control. We use the spores of Bacillus pumilus. So that is about the ionizing radiation. And the non-ionizing radiations like UV rays and infrared rays are having more of an application in case of sterilizing air disinfection or like water disinfection. So those are the situations where we use the non-ionizing radiations. Now coming to the next physical method and the last physical method of sterilization, that is filtration. So heat method is over, radiation method is over, and the last one is filtration method. And what are the uses of filtration? When you filter something, you use a filter. The filter will be having some pore size, and anything which is bigger than this pore size will be retained in the filter and the rest of the thing will come out. The filtrate will be free of those things. So we use filtration to remove bacteria from heat labile liquids. Those liquids which are heat labile, you cannot heat it, like serum, sugar, antibiotic solutions, vaccines and all. We do a filtration method so that you can filter out, you can remove the bacteria from such solutions. And the very important application of filtration we all are familiar with is water purification, right? We use filters at home for water purification. So those are also basically filtration. And air purification, we have certain special filters for air purification which you assemble in the hospitals, in ICUs, in theaters, surgical operation theaters and all. We use this air filters so that the air which come inside the room will be filtered and free of microbes. In water bacteriology, you have application because when suppose you suspect that a particular water is contaminated with certain microbes, you want to know what kind of microbes are present in it. So you subject this water to filtration. So the filter will be retaining those microbes so that you can culture it easily. So that is the application of water bacteriology. And also we do it to obtain bacteria-free filtrates for virus isolation. Suppose you're going to isolate virus from a sample. You want to remove all the bacteria from that sample. For that also you can do filtration method. So the most important application is for removing bacteria from heat labile liquids, water purification, air purification, water bacteriology. You have basically two types of filters, depth filter and membrane filters. Depth filter means the entire thing will be used as a filter. It's not just a single surface filtration. The whole depth of this material, if there is a filter like this, the whole depth will be doing the filtration. So when the filtration is being done, the entire material will be doing this process of filtration. So that is not being used nowadays, but you can remember some names for the purpose of writing an exam. Example for depth filters, there is candle filter, asbestos filter, and sintered glass filter. So these are the filters that we used before, long before. An asbestos filter, there is one name called seeds filter. It's not being used now. And candle filter, you have different kind of candle filters, and one commonly used one is called as Chamberland filter. So these filters, you can remember the names for the purpose of exam: candle filter, asbestos filter, and glass filters. But these are not used now. What we prefer now is membrane filters. So what are membrane filters? You can see here a single membrane which is being kept here. So in this, here the filter is kept. So this will be looking like this. If you look this membrane under microscope, you can assume like this. The filters will be like this. There will be pores in between the filters, and all the things which are bigger than the pore size of the membrane will be retained on the surface of the membrane, and the rest of the thing will come through the pore to the as a filtrate. It will come out as a filtrate. So you can see the filtered fluid that is coming out will be free of all the microbes which are retained on the surface of the membrane. So what are they made up of? They are made up of cellulose esters, nitrose cellulose esters. They are porous because they contain pore. And depending upon the pore size, they can retain certain organisms. Generally, the pore size that we use in most of the membrane filter is 0.22 micrometers. We know most of the bacteria are around 1 to 2 micrometers in size. So all those microbes which are bigger than this pore size will be retained on the surface of the membrane. And how do you sterilize this membrane? See, this membrane which we use for filtration should be sterilized, and that is sterilized by autoclaving. So membrane filters are being used. It's very good, better than the depth filters. So what they do is just surface filtration. And the last kind of filters that you should learn is air filters. I told you air filters are being used in our hospitals. We have two types of air filters. One is HEPA filter and the other is ULPA filter. And what is HEPA? HEPA is high efficiency particulate air filter. High efficiency particulate air filter. That is HEPA. High efficiency particulate air filter. So these are being assembled to the air inlet valves, inlet areas, and it is usually of a pore size of 0.3 micrometer, so that the air that comes towards a room, inside a room, will be filtered, and all the microbes will be filtered by the HEPA filter. So you get a microbe-free filtered air inside the room. This is specifically important in case of operation theaters where you perform the surgeries. You need a filtered air. And when it comes to ULPA, ultra low particulate filters, in that case, the pore size is again small, 0.12 micrometer in size. So it has got a better filtration capacity. HEPA filter is supposed to filter out 99.9977% of the microbes which are present in air. So just remember the name of the air filters: HEPA filter and ULPA filters. So with that, we have completed the physical methods of sterilization. Being heat methods already covered, radiation methods, filtration method have covered. So we'll just see a pyramid showing the resistance of microbes so that we get an idea what kind of method of sterilization or disinfection you should choose. So when it comes to sterilization, sterilization comes under the top priority, because they are the best method. They can destroy spores, S4S, that's how we remember, right? Sterilization can destroy spores. So bacterial spore is supposed to be the most resistant form of microbe, but there is something else called as prions, which you will learn later. Prions are actually even smaller than viruses. They just have a protein material. There is no nucleic acid. Infectious proteins are prions. They are highly resistant to all kind of sterilization disinfection methods. We need special methods for them. But in general, among all other microbe forms, spore is the most resistant form, and to destroy spore, if you are suspecting spore in a sample or a material, you have to go for a sterilization method. You can't take risk. Now, when it comes to TB bacilli or Mycobacterium tuberculosis, you require a high level disinfection. We'll learn the chemical disinfectants in a coming class, in which you can see what is high level disinfectant. Now, if you're dealing with non-enveloped viruses which doesn't have envelope, like your hepatitis A virus which doesn't have envelope, in that case you can go for a high level or intermediate level disinfection. And what are the most vulnerable organisms which can be easily destroyed? Those are the normal bacteria, fungi, and the enveloped viruses. So remember, viruses with envelope are more susceptible to disinfection methods than non-enveloped viruses. So the one which comes under the bottom of the pyramid is our normal bacteria like E. coli, staphylococci, normal fungi like candida, mold, and non-enveloped viruses like the coronavirus or the HIV virus. And the next level of pyramid consists of the non-enveloped viruses like hepatitis A and polio. And the next level is Mycobacterium tuberculosis, which is more resistant. Then comes the bacterial spores. At the top of the pyramid, it is prions. So, depending upon the kind of organism that you're going to deal with in an article, you have to increase the level of disinfection or sterilization. There is a classification called Spaulding's classification, which is important in your clinical practice. You have to classify the items to be sterilized based on the risk that it poses to the patient. You can divide it as critical, semi-critical, and non-critical items. I'll start with the non-critical items. Everything which comes in contact with the patient's skin, just like the bell of a stethoscope or the BP cuff or the bedpan, all those things come in contact with the patient's skin, intact skin of patient. Such kind of articles, if you want to sterilize or disinfect, you can just go for an intermediate or low level disinfection. You don't have to go for a bigger kind of method. And when it comes to the certain other items which can touch the mucous membranes or touch non-intact skin, the most classic example for a semi-critical item is a endoscopes. All kind of endoscopes which is going into the mucous membrane, respiratory or GIT or whatever. So endoscopes are supposed to touch the mucous membrane of the patient. They are considered as semi-critical items which require a high level disinfection. You can't choose a intermediate or low level for them. They need at least a high level disinfection. And what are critical items? All the items that are going to get into a sterile site like the blood vessel or into your visceral organs. So all the surgical instruments, the intravenous catheters, the IV lines, all those things are entering the sterile sites. For such kind of articles, you cannot go for any lower method. You require a sterilization method. So for critical items like the IV cannulas and surgical instruments, it is sterilization. For semi-critical items like endoscopes, it is high-level disinfection. And for non-critical items like the BP cuff or stethoscope, bell and all, it is just a low-level or intermediate level disinfection is enough. And this classification is called as Spaulding's classification. This can be asked for your MCQ exams. So this also you should be familiar with. So with that, we have completed all the physical methods of sterilization, and what is left is chemical disinfectants, which will be dealt with in a separate class. And please go through the PYQs, the previous year questions. All these were previously asked, and I hope all these points are clear. I have put one more slide for sterilization control as a summary, because when a question was asked as sterilization control as 5-mark question. For that, don't just write a single method. You have to write the control for all the methods. Like hot air oven, what is the biological control? Bacillus subtilis. Autoclave, Bacillus stearothermophilus. Radiation, it is pumilus. So pumilus, you can change P to R, right? That's how we remember. Cumulus is the control for radiation. And for ETO, ethylene oxide sterilizer, which we have not learned here because it comes under chemical method. ETO, the thing we use as a control is Bacillus globigi or Bacillus atrophius. And these are all MCQs also. And physical method, we need a thermostat. And this chemical method, you have the tubes and tapes. So these are the sterilization controls that we use in physical methods of sterilization. So this is also a previously asked question, and with that we wind up the physical methods of sterilization. Thank you.